So I'm Natalia. I'll be teaching you today about chemical reactions. I'm a protein biochemist and structural biologist from the Rockefeller University here in New York City. And before we start learning a bit about chemical reactions, I just wanted to say that I am so glad I could be here to teach you all today. And I'm going to try to make this experience a little bit interactive. So I'll be asking some questions, and feel free to answer the questions by unmuting your mic, or you can put it in the chat. And or you can raise your hand. And so I welcome questions. So if you have a question in the middle of the presentation, you can just raise your hand, or you can unmute your mic, ask a question, or ask a question in the chat. And so I'll be telling you guys a little bit about myself to begin with, but just a little warm-up question, just to start exercising your minds. I want to ask you guys a question about your favorite ice cream flavor. So my favorite ice cream flavor is mint chocolate chip. So what is your favorite ice cream flavor, if you have one, if you even like ice cream?
I personally like sorbet more than ice cream, but strawberry is my favorite.
Yeah, I like strawberry too. It's really refreshing. Does anybody else want to share?
I like mint chocolate cookie and mint chocolate chunk.
Oh, so you're like me.
Well, somewhat.
Somewhat, yeah.
I like chocolate, I guess. There are a few other things I like.
Yeah. I mean, yeah. I mean, there's so many different flavors. I'm pretty sure that my favorite flavor changes, you know, with by the year. So not
Yep.
often we have the same flavor.
Flavor
I also
or ice cream?
like sorbet more than ice cream. But
Yeah.
if I do have to choose, I'd choose mint chocolate chip or cookie dough.
Oh, cookie dough. You know, I'm from Australia, and we didn't have cookie dough ice cream when I was growing up. So when I came here, I thought it was really amazing. But okay, so I mentioned to you guys that I am a structural biologist. So I'm going to share my screen really quickly just to tell you guys a little bit about what that means. Actually, first of all, I want to ask you guys a question.
Oh, there's a poll. That's
There's a poll question. So does anybody know what a protein is?
Yep.
Okay, just waiting. It's my first poll. So Adelis, do we end the poll?
How did you do that?
How did I do that? How did I ask you a question?
Yeah.
I have some controls here that allow me to do it because I'm the host of this presentation. Well, the co-host with Odelis.
I didn't know you could do like surveys on Zoom though.
Yeah, you can. I mean, I just learned about it last week. So, all right, so I'm going to end this poll.
So I just shared the results.
Oh, okay. How do you see that?
So 86% said yes, they know what a protein is.
Okay, that's great.
14% said I've heard of it, but I'm not sure what it is.
That's okay. So, proteins are found inside or on the surface of cells. And so they can be bacterial cells or viruses or on the surface of your skin or inside your skin or brain cells. So any type of cell contains proteins. And these proteins perform important functions in the cell, which helps the cell to survive. So I'm going to share my screen with you really quickly. So can everyone see that?
Yep.
Interesting.
So, oh, we've done that. So as I mentioned, I am a protein biochemist in structural biology, which means that my job is to study proteins. and to understand what those proteins look like. And so to make that a little bit more clearer to you, when I was in grad school, I studied this bacterium here, and this bacterium is called Streptococcus pneumoniae, and it causes the disease pneumonia, which is the disease of your lungs. And so when you look a little closer into these Strep pneumoniae cells, bacterial cells, you see all this murky stuff. And in that mixture of murky stuff is where the bulk of the proteins are that this disease-causing bacterium needs to survive. And when you take an even closer look at this murky stuff inside these cells, this is what you see.
Oh,
And
wow.
so all of these different colored objects are what is found inside a bacterial cell. And your cells probably look very similar to this, sort of, if not a bit more crowded. So
Are they actually that colorful?
Well, no. This is just helping people to visualize all the different proteins
So
and objects
colors
inside
so
the
that
cell.
you can easily tell them apart and visualize them, though not technically nothing is actually colored since color is an illusion.
Well, we'll be talking about some proteins that actually do have a color a little later. But here you see like proteins, and you see genetic material such as your DNA. Well, this bacterial's DNA, bacterial cell's DNA, and some RNA. And so this is what's inside a protein. And so what protein biochemists want to do, or what they do, is they study each of these individual proteins. They study their function. And so the reason is each of these proteins are in such close contact with other proteins, they actually work with other proteins. So just imagine each of these circles is a protein, and each of these lines mean those proteins interact with each other. And so protein biochemists study the interactions between proteins to understand how bacteria survive, or how particular functions inside bacteria help them to cause disease.
So what are proteins in general?
So proteins are essentially objects inside the cell that perform essential functions in your cell to help your cell survive. So it's like the machinery of your cell are performed by proteins. And so what you can do, so what I did as a structural biologist is I got the structure of a protein. So this is the protein structure that I solved in grad school. And so this structure allowed us to understand how we can use this protein, which is essential in Streptococcus pneumoniae, how we can use it to potentially treat individuals that have pneumonia by targeting this protein inside the bacteria.
I didn't know pneumonia was caused by proteins.
Pneumonia isn't caused by a protein, it's caused by a bacterium. And inside that bacterium are thousands of proteins that are essential for the bacteria to survive. And so the idea is if you stop one of those essential proteins that the bacteria needs to survive, you can kill the bacteria and stop it from causing disease.
So, question, I don't know if you can hear me, I have very bad audio, but for the time being, so you're saying, for example, the life function in this bacteria is made up out of small chemicals called proteins, which consist out of ATGP particles. I know this because of prior knowledge, but it's essentially like life code. Essentially, I don't know if life code would be.
So you're talking about DNA?
No, not DNA, but like you said that proteins are the basic building parts of that bacteria. So like if you were to turn one protein off in a bacteria, would it turn off the special function? Is that what you're saying?
So proteins perform essential functions in bacteria to help them survive. And so if, for example, one protein made a particular nutrient that a bacteria needed, right? It was essential in manufacturing a particular nutrient inside a bacterial cell, like an amino acid, for example. And so if you stop that protein from being able to perform its function, then that bacteria can no longer manufacture an essential amino acid that it needs. And so without that amino acid, the bacteria could no longer survive.
Oh, I'm sorry with the ATGP stuff. That was later on in the last.
Does that make sense?
Confused for a second.
Okay.
As for the question that I was asking, thank you for giving me the answer. This was a truly enlightening inquiry.
Okay. Would you be able to repeat that question for me? Sorry.
No, you answered it. I was
Oh,
saying,
I answered it. Oh, okay.
thank you.
Oh, no worries. No problem. Okay. So, so now I'm going to move on a little bit. So that's pretty much what it means to be a protein biochemist and a structural biologist.
And
so
sorry.
yeah, go for it, Cameron.
How many proteins are in like one cell usually?
Oh, whoa, that's like, who knows? It's like, it could be hundreds of thousands upon thousands of proteins inside your cell.
Okay, then.
That's how complicated human cells are, or any cell for that matter is. They need a lot of things to survive. Okay, so now back to chemical reactions. So before I get into chemical reactions, I'm going to ask you guys a series of questions. So my first question is, so there are a lot of objects that we can see with just our eyes.
Yes.
Could anybody name me an object, just one person, just an object that you can see with your eye
I'm the computer.
that's taller than your teacher?
Well, I can see my bed, and it is taller than my teacher's.
I'm pretty short. There's probably a lot of things to me.
Okay, so how about an object that is smaller than your fingernail?
Well, I guess.
Ooh, this.
What's that, Cameron?
An eraser.
An eraser. How about something even smaller than an eraser?
A piece of dirt, I guess, like or a grain of sand.
That you can see with your eye. Yeah, a grain of sand, right? An ant, a little bug. So these things are really, really small. And so the limit of what you can see with your eyes is about 1 millimeter. And so if you wanted to see something, so for example, one of the largest things you can see, like one of the largest living organisms on this earth, is General Sherman, which is a sequoia tree in the Sierra Nevada mountains. You can see mice, you can see bugs. But what if you wanted to see something that was 1,000 times smaller than an ant? You need a special microscope. You need a light microscope. And so can anybody tell me what it could be that is 1,000 times smaller than an ant that you might want to see?
I think tackling the way first thought ladybug.
Sorry, Sky, what did you say, Sky?
I'm guessing, but I said
What about a sperm cell? A sperm cell.
A cell? Yeah, yeah, a cell, yeah.
Maybe a super, super, super young fetus.
Yeah, like, okay, yeah. So we can see many different things that you can
Those
see
are
with a light microscope.
Those are actually, I think, still somewhat visible. You just can't see through people's stomachs.
Well, you can't see them also with your eyes. You need some assistance. So you need a light microscope because they're 1,000 times smaller than the smallest thing you can see with your eye. So for example, you can't see an individual red blood cell with just your eye. You need a microscope. You can't see an individual bacterial cell that's over here. You need a microscope. You can't see an individual skin cell to see all the parts inside the skin cell without the aid of a light microscope. So these objects are about 1,000 times smaller than a bug.
Hello, I'm sorry, let me turn up my audio so you can hear me. But what I did wanted to say was that I think that what was I gonna say? Was something that pond water, I mean algae, is like small enough to be seen with a light microscope. After all, we have seen algae in class.
Yeah. Yeah, you can see algae with a light microscope. So if you wanted to see something that is 1,000 times smaller than each of these, smaller than a bacterial cell, smaller than your single cell, smaller than...
I have a question.
Sure.
Is it possible for something to be so small you cannot even see it with a microscope?
Yeah, so we're just gonna, yeah, it is. So for example, to see things like, okay, some of the smallest things, you need special microscopes and special techniques. So for example, a virus particle, you can't see it with a light microscope. That is about 1,000 times smaller than your cells. Then
DNA.
you have
DNA.
proteins, which are 100 times smaller than a virus cell, which you cannot see without these special microscopes. And then you have the even smaller things, your DNA, that is even smaller than a protein, that you have to see with these special microscopes. And so what this is all getting at is that often we can't see chemical reactions from occurring. They're performed by proteins, they're performed with the aid of proteins and other chemicals in our atmosphere that are smaller than DNA. But there are some chemical reactions that you can see. And so here are a couple of chemical reactions that you're able to see. So for example, here you have a colorimetric reaction. So two chemicals mixed together, they form a color, so you can visualize it. And here on the right, you have the phenomenon of bioluminescence, where you have these these organisms inside the seawater that perform a chemical reaction that produces light. And so that's why you see that they're blue here. But today, what I'm going to be showing you are chemical reactions that involve proteins. And the protein that we're going to be using to teach you a bit more about chemical reactions is the protein, green fluorescent protein. So this protein actually fluoresces green because of this part inside the protein called a chromophore. And so that's how the protein got its name, because it's green. And so this protein is one of the most widely used proteins in science because it allows you to visualize where things are in cells. And so I have another question for you. Let's see if I can, oh, Dallas, I can't. Oh, here it is. I have a poll question.
Question: green fluorescent protein.
Where do you think,
If it were found on land,
where do you think GFP is found?
I'm sorry.
So is it found in the land or in the ocean?
Go for, I'm not gonna say well.
Yeah, 50% who voted, so I'll give you all a few more seconds. So you have a 50% chance of getting it right.
Okay, 50.
Now I'm wondering. Hmm.
Oops, I might have given the answer away. So everybody,
Everyone voted who wanted to vote. I'll close it in 5, 4, 3, 2, 1.
all right. Okay, so what do people say? I can't see.
Awesome. So 100% of you all said it was found in the ocean.
Yeah, it's found in these jellyfish, a special species of jellyfish called Equaria. I can't say that properly, Latin, Victoria. And as you can see, it's green because in its cells it has this protein that fluoresces green.
Actually, I had to do a little deduction together. But, the only thing that would probably have green fluorescent protein is plants. And I don't know any plants that can turn a bright green. I'm pretty sure that animals don't have this other than seawater animals. So I went with the ocean. I mean,
But
it's
yeah,
a logical choice, assuming what we know is true.
I'm
Yeah.
really sorry. I completely forgot. I
That's
thought it was
okay.
next period, not this period.
That's
That's
okay,
okay.
Daniel. We're just gonna catch you up, okay? Just sit
Excuse
tight.
me. Excuse me.
Yeah, sure. Hi,
Can you
Jonah.
say the specific topic we're on? Because I've got a question, but I'm not sure if it's on topic or just on topic with something you brought up.
You
But
could, any question. I mean,
so
we're talking-
oh, and I just want to make sure this is on topic. So can literally the topic we're talking about?
So we're talking about chemical reactions involving proteins.
Okay. Yeah, my question would be like, since we were talking about light and microscopic stuff,
Mm-hmm.
but do you want me to ask it?
So we were talking about light and microscopic stuff just to introduce you to things that you can't see. And a lot of times
Yeah,
things you can't see.
exactly. That's
Yeah.
why this would be off topic.
Yeah. So, and this is the protein that we're going to be using in the experiment that I'm going to show you later. That's going to help us to see chemical reactions in action. So, so I'm just giving you a brief introduction of this protein before we start working with it. So, so this is one of the most widely used proteins in science, and it was considered such an important discovery, and it's so important and life-changing to the field of science that the people that discovered this protein were awarded the Nobel Prize, which is one of the greatest prizes any scientist can be awarded. And so people recognized how important this protein was to scientists, and it's incredibly important to this day. So before we move on to chemical reactions, I just wanted to make something clear. So I mentioned before bioluminescence, and I mentioned before fluorescence. And so I want to ask you a quick question. Are bioluminescence and fluorescence the same thing?
So we have about 60% who voted. So those who would like to vote, I'll give you a few more seconds. So hopefully everyone can see the poll where it's asking a yes or no question.
Oopsies. Okay. So has everyone voted?
Yeah, I voted.
Okay.
Awesome. So I'll end the poll and I'll share the results.
Cool.
So we have about 80% that voted no.
It
And
says
It says
100%
100% voted no
It does
and
say
20%
That's
voted
not
yes.
possible.
but that's not possible. So I think Zoom has an error. But it's 80%, it seems like, voted no, and 20% voted yes.
Is it possible to change your answer?
Oh,
No.
maybe.
Or maybe some people pressed both of them
Oh,
by
yeah.
accident and that glitched it?
Maybe, yeah, maybe Zoom has a glitch where you can log into the poll again and change
Yeah.
your answer.
That's probably what happened. But majority said no.
Okay, well, the majority are then correct, because bioluminescence, as I mentioned before, is actually a type of phenomenon called chemiluminescence. And so that is where the chemical reaction produces light, but it's called bioluminescence because a biological entity is creating the light. So in this case, over here with the blue light, it's being caused by dinoflagellates, which are inside the water. And as the water gets essentially agitated by the person's hand or by the waves crashing on the shoreline, you can start to see the light being produced. And on the right side here, you see all of these little bright lights happening here, just twinkling inside this forest. And that's being caused by this bug here. And this is also another case of bioluminescence. So just quickly, does anybody know what this bug might be?
Firefly.
Exactly, it's a firefly.
And also, is bioluminescence basically just when a living thing makes light?
It's when a chemical reaction a living thing produces a chemical reaction.
Can I ask, does that mean, in what way? Because, can you be more specific? Because if we're rubbing sticks together and make a fire, technically that would be a living thing, making the light.
Not really.
When the proteins make light.
It's not really because it's the use of tools making the fire. Also, it's two sticks that you are using that made the fire, not necessarily your own chemical fluid. So it wouldn't be
But technically, it's the sticks making the reaction.
Yeah.
Yes. So there's two ways to make
In-
light, Jonah. So
What?
so there's a couple of different ways to make light, and so we're gonna go through that. But like, yeah, you can make light, physically, and then you can make light how these animals do it internally using chemicals inside their body and proteins that help them create the light. So these are chemical reactions that are inside the animal that produces light. And so, yeah.
amazing.
So there's music. So then okay, thank you.
So this might make it a little clearer for you. So bioluminescence is a type of chemical reaction that's referred to as a synthesis chemical reaction. And so what that means is that you have one object plus another object, which synthesizes this new object, which is AB.
Excuse me?
Sure. Hey.
That would mean A plus B equals A times B.
This isn't a standard mathematical question. This is a chemical reaction, so it's a little bit different.
Yeah, but
But basically, it means when you put one thing with one thing, it makes
can you
another.
give some constructive
It could be
on
equal to
precision?
2. 2 plus 2 is 4, and 2 times 2 is 4.
Okay, so let's go through this chemical reaction here and try to wipe away mathematics from your mind for a second. So what A is, for example, is a compound called luciferin. So it's a chemical compound. And then in the presence of oxygen in the atmosphere, this protein called luciferase combines the two together, helps combine the two together to produce this new compound called oxyluciferin. And that oxyluciferin releases light. So this is essentially what's happening when the dinoflagellates and the fireflies are producing light. Is this luciferin reacting with oxygen, which is helped by this protein called luciferase, which produces oxyluciferin, which then releases the light.
So so does that mean light is considered?
Have I missed something?
Wait, can you say something? Does that mean light is considered physical matter rather than energy?
Well, light has energy, but it's
but is light considered physical matter, energy?
It's a physical property that has energy.
Let me give you a little.
So it's like plasma, as in the combination.
It's sort of like, imagine this. So luciferin plus O2 equals a bit of energy, which creates light, and also oxyluciferin. So the bit of energy turns into light because that's the way it was exhorted. Because you cannot just keep the energy inside. You have to let it go because energy is made from chemical reactions.
I think it's basically when like there's one thing and another thing, and then they combine in a chemical reaction and create something else.
So yeah.
So,
So this.
Wait, let me ask this. Is light then, does that mean it's made of like matter?
No.
Well, it's made up of like rays and particles and stuff.
So it's yeah. So light is high energy. Sunlight is something that is visible because it's made up of a component of many different types of light, right?
But so I'm asking, here is the light made partly of matter?
And light is a type of radiation. Like there's lots of different types, like infrared and microwave, that we can't see but are still there.
Yeah.
That's not what I'm asking.
Jonah, light is an energy form, it's a form of energy. So even if light is coming from the interaction of matter, light itself is energy. It's something that gets measured in photons. It's energy.
Okay, so there's no matter. So
It
gets
what
measured in our
happens
prep.
to the matter?
It interacts with matter.
Yes, it's interacting with the matter, or it's coming from an interaction of matter, but it's itself being given off as energy.
It's basically just energy coming from the sun in a way,
Well, it's coming from this
energy
chemical.
can't be created nor destroyed.
or a light or whatever.
Yeah.
Energy can be created nor destroyed. So how does a chemical reaction, a combination of two?
It's got to do with the protein, the compound luciferin. It's a special type of compound which has the ability to emit photons.
Yeah.
And so that's how it produces the light.
So wait, is light like a what? Say, is light like plasma then?
Oh, I don't really know.
Plasma is a type of matter.
Plasma. Plasma.
It's basically matter with electric charge.
It's not.
No, I'm not.
They're different.
Yeah.
So
Give me a second.
okay, so this is fluorescent. So this is two types of fluorescence.
So please.
So fluorescence is a physical property not caused by a chemical reaction. And so what's happening here is that you have this chromophore in the protein which absorbs light at a particular wavelength, and then it emits light. And so if your chromophore is a green chromophore, it'll emit green light. If it's a red chromophore, it'll emit red light. So fluorescence is a physical property not caused by a chemical reaction.
Wait, so it's basically just like something that happens that's not caused by a chemical reaction?
It's not caused by chemicals, yeah. So
It's
this
like-
chromophore.
So like this. Okay, so like think of it like a stat. Is it for fluorescent? Glow stick, yes. Chair, no. Like that?
Sorry, I couldn't quite
But wait, so
understand.
like what is it, like in simple terms?
Is it like a stat? Like
What's a stat?
glow stick is for fluorescent, but a chair is not?
Yeah.
Oh, I thought I was incorrect.
It's so this chromophore has the ability to emit lights of a particular color depending on which light it absorbs.
Oh wait, is it when a thing emits light without a chemical reaction?
Yeah, it's when, so fluorescence is not caused by a chemical reaction but has the ability to emit light.
And does it have to be a
And
living
that's
thing?
fluorescence. It has to be a functional thing. I mean, this is a protein that has the ability to do that. It will do that when it's not even inside a cell. So it doesn't have to be a living thing. It just has to be a functional thing.
Cool.
So a protein will be able to fluoresce. It's like your glow stick will be able to fluoresce or whatever without being inside a cell. Yeah. So
Wait, so it's basically like the opposite of bioluminescence.
yeah, because it needs other things around it to be able to perform its function. Like it can't just put it on its own. So like this protein can create this color on its own. It can fluoresce on its own. Whereas bioluminescence, it requires
work.
Sort of like it required the luciferase enzyme to help bring together the luciferin and the oxygen to produce, the oxyluciferin in the light. Whereas here, it just needs light. And so when it absorbs light, it emits light of a particular wavelength. So here, it emits green light because the chromophore absorbs
I think
blue light.
By the way, do you know how thankful I am for having you here today? You have put in a lot of energy into doing this, and I'd like to thank you.
Oh, thank you, sir.
It's invisible, but it's there.
Oh no, we're going to start getting to the visible part. Okay, so today let's see how we're doing for time. I think we're okay. So I'm going to do 2 examples of chemical reactions that involve proteins. And so the first chemical reaction is going to be a protein test. So the chemical reaction will occur and it'll be a color change. So you'll be able to see the chemical reaction if protein is present in the solution. And then the second chemical reaction will be the effect that different environments have, different chemical environments have on fluorescent proteins. Okay, so let's see if I can get this started.
Natalia, yeah, I was just going to say, stop sharing your screen.
Okay, so, okay. So I'm gonna see, how do I make myself enlarged? Because at the moment, you're the largest thing I can see, Odelis. Okay, I don't know if you, oh, okay.
Oh, I was on mute, sorry. Are you going to use the camera that you're looking in right now?
Yeah, I'm just gonna use my.
So I just spotlighted the video, so now everyone sees you.
Oh, okay, cool. Okay, so I'm gonna try to focus on my workbench here.
So now she's going to show you what the reactions look like. So she's at her bench taking her supplies out. So she has some tools that's going to allow her to add and remove liquid.
Ooh, this is cool.
This is very cool. To ask a question too. Not too much of it.
I hope everybody can sort of see this.
Mm-hmm.
So I have, to start with, I have my proteins. So I have 2 proteins here. See if you can see these guys. You guys see them?
I bet one is like the red fluorescent and the other is one is the green fluorescent and the other is.
But if proteins are in cells, then how did you get them out of the cells?
Oh, okay. So this is the green fluorescent protein. This is the red fluorescent protein. And so the way that I got them out of cells is I cloned the gene into a bacterial cell. I don't know if you can see this.
Kind of.
Can see it.
And so on this plate is individual bacterial cells that I cloned the gene into. And so when I cloned the gene that produces green fluorescent protein into these cells started to produce that green fluorescent protein. And then I was able to extract the protein from these cells.
That's cool. Wow.
And that's what you see here. That's the extracted green fluorescent protein from those bacterial cells. And so pretty much you can clone any gene, pretty much any gene, into specific cells, and those cells can start making those proteins, and then you can purify those proteins from those cells. Okay. And so that's our green fluorescent protein.
You don't know, and it's also possible to actually clone animals, like Wendy.
Like a full animal?
Uh-huh.
I mean, yeah, there was Dolly the sheep. There was a sheep that was cloned from another sheep.
You could pay like I think $20,000 to clone your dog.
Mm-hmm. Really? I didn't even know that.
Yeah, you can clone them.
I crazy, right?
What do you mean clone them?
Get a puppy with the exact same genes.
Well, what would it actually do though?
Very risky. And it undertakes a lot of dangerous testing. I don't know if it's a good idea.
Illegal in the US.
And that's kind of a waste of money.
So wait, it just comes out with like an actual alive dog?
Yep.
Yeah.
That's creepy.
Well, it's not like a machine or anything. It's do you know how IVF works? It's you can take a petri dish with like an embryo in it or whatever. It injects sperm into that petri dish and it can grow an embryo. And then,
okay, guys.
It's true in vitro.
Focus, guys.
Focus. So we've got these 3 tubes here, and you can see that tubes 1, 2, and 3. And so we're going to use this protein assay, this dye here, And this dye, when it comes in contact with protein, it'll change color. And so that's a chemical reaction. And so we're going to see which of these tubes has protein in them. And you can already tell that some of them have protein in them, but we'll see whether the dye picks up those tubes that contain the protein. Okay, so I'll just get some tubes out. Ooh. Okay, so we'll do a control. You always have to perform a control. And then we've got tube 1, 2, and 3. And so you sometimes when you a lot of the times when you're performing experiments, you want to know how much protein is inside, how much protein you have, because you want to be able to reproduce that experiment. It's like a recipe. So if you don't know how much sugar to add in your recipe, you won't be able to reproduce that recipe very well. Okay, so let's do 1 in. Okay. All right.
I think that like cloning procedure is pretty. I think that this way is sort of efficient. This way of doing tests is really efficient. I support this. With fluorescence, of course, it's pretty good.
And so I'm going to do a control tube that just has water.
Well, that's pretty smart, because then we know the difference between what would happen if we'd never put this protein in at all, and what's the difference?
Exactly. So whenever you perform an experiment, you always have to have correct controls, because someone may ask you, how do you know what you're seeing is real? Okay, so now I'm going to add my dye. And we'll see what happens. All right, so here's my dye. It's brown. So I'll add it to the control tube. Okay.
Oh my god. Oh my god. Oh my god.
No?
What?
Is everything all right over there?
Sorry, I just thought of an experiment I really want to do.
Oh, yeah?
Yeah, so you see, there's this thing called an ecosphere, which is basically an like an ecosystem inside a little aquatic ecosystem, self-sustaining ecosystem inside like a
Oh my gosh, the water was
sphere,
the
a
only one
glass
that's brown.
sphere, aquatic one. All you have to do is give it some light, and by resting it on the windowsill.
Oh, yeah.
All right, Jonah, let's take a look at this experiment.
Oh yeah, I'll
Oh
show
my
you
God,
the
I don't
experiment.
think the control is in her hand.
This control is this one here, sorry. So we'll just, it's hard to sort of like, so the control is this one. It's in my hand over here. And then let's see if I can.
Wait, you sure the food coloring is going to affect the control?
So this is the control, and then you have tubes 1, 2, and 3.
So why are they different colors?
Oh, but Cameron, think about this. The water just diluted the original thing. And so because the original thing was tinted a certain color, the other ones actually had a chemical reaction, while the control didn't. Because
So So the
Okay.
water,
What was the original color of the
so
dye?
this is the original color of the
Oh,
dye.
that's
So the
the
water, so the water I added 400 times more dye than water. So the water would not have been able to dilute the control reaction. So this is pretty much just the color of the dye with no protein. However, when you start adding protein, you start to see a color change. So the dye, so is no longer brown, but starts to become blue, right?
Did I launch the poll?
Wait, so the blue ones are fluorescent proteins?
So this has nothing to do with fluorescent. This is a chemical reaction that's happening between the dye and the protein.
The protein. So the 1 and 2 have protein in it. Does 3 have a protein?
So how about we launch a poll question really quickly to see which tubes you think contain protein in them?
Tubes or tube?
1 tube.
Which
So
one
pick one.
contains the most protein?
So did you guys, yeah.
By the way, this is a multiple choice question, or it looks like it's a multiple choice.
Yes, you only get to pick one.
You get to pick one.
Oh no, I picked twice.
You were able to do that? Okay, Zoom floor.
Yeah.
I chose 1 and 2.
You can only choose one. Choose one.
Wait,
why do I
one
have
on the
to choose
multiple
number
choice?
1? Why can't I choose number 3?
Because there's
You
only one
Choose
right
number one,
answer.
but you only get one choice.
You get to choose one choice on the multiple choice.
Oh no. Okay, I'll just.
Is everyone in?
All right, so I'll give a few more seconds. Anyone else
Oh,
wants?
now I understand. Okay.
I made a mistake. Oh gosh, I can't
Like
fix.
2 of them have water?
So her control tube had water. So the one on its by itself, and then 1, 2, and 3 had a certain amount of protein in each of them. So she did a chemical reaction to see which tube has the most protein. So the question is, which tube do you think has the most protein?
So there was a question, which tube has protein and then which tube has the most protein? So did you guys get both questions?
Oh no, I just did
No,
which
I
tube
only
has
got
the most
one.
protein.
Oh, so one of the questions was which tube do you think, which of these tubes do you think contains protein, if this is the control here?
I can ask that question quickly.
Yeah, so we'll ask that question. So which tube do you think, which of the tubes contains protein? That's going to be a multiple choice question.
Well, a lot of them have protein in it.
Yeah, so just, we'll see the results of the poll.
Wait, no, I was wrong. I got it wrong. I didn't mean to get it wrong though.
And so this is the control here.
Misclicked. Darn it.
Okay, so does everyone answer?
Wait, so I don't understand what happened. Like, why are they colored differently?
Okay, so we'll answer the first question first. Which tubes do you think contain protein?
So sharing the results. So the majority said tube 1 and 2, and the close second was all tubes.
All right, so yeah, all tubes contained protein. As you can see, there is a slight difference between tube 3 and the control. And so the second question was, which tube do you think contains the most protein?
Wait, so what happened?
Yeah, so when we answer which tube contains the most protein,
it makes it look different. There are certain different amounts of protein between each thing.
yeah.
So now the next question is, which tube out of 1, 2, and 3 that Natalia has in her hand do you think has the most protein?
But I thought 3 of them were protein and 1 of them was water.
Yeah, so 1, 2, and 3 has protein.
This one is the control reaction that has water. And so at the start, we didn't know which of these tubes have protein. And so we're basing this all on the chemical reaction that's taking place that you can see before your eyes, which is the color changing from brown to blue.
Why is 3 gray?
Oh,
Has everybody answered that question? The second question about which tube has the most protein?
of people voted. Anyone else want
Okay.
to vote? Okay, we got one more vote in, and I'll close the poll. So there's a tie with 2 and 3 having the most protein, with 2, 1 coming in a close second.
So 2 and 3 have the most protein. Is that what was that
Yeah,
it?
that's what was chosen.
Okay, so if this tube, this control tube here just contains water and has no protein, this has the least amount of protein in it.
I know.
And then the one where the greatest chemical reaction, the greatest degree of chemical reaction has occurred is the one where it is the most visibly different to the control tube. So number 1 is the most visibly different to the control tube.
I'm sorry, my brightness is very low, so 1 and 2 looked like the same thing to me.
Oh, that's okay. That's all right.
Why is 3 gray though?
Because there's just a small color change happening. So it's the result of there being some blue formation, which makes it a little bit gray and less brown.
Oh, so there's not that much protein in number.
There's not that much protein in number 3. There's more protein in number 2, and there's the most protein in number 1. I'm sorry, Samuel, that you can't really see because of the resolution. But
wait, so it was brown dye originally.
Yeah, it was this color originally. And then in the presence of protein, more and more amounts of protein in the solution, more of that chemical reaction can occur and produce this blue color.
Like the more protein in it, the more blue it will be.
Exactly. And so I'm just going to go back to the presentation real quick, just technical stuff. So that chemical reaction is a synthesis reaction that's happening. So you have the protein, and you put the protein in a solution with a particular dye that only binds to protein. And that dye, when it binds to protein, it becomes this protein dye combination which releases a blue color. And that's just specific to the dye. The dye, when it binds to protein, it releases a particular, it changes state and it produces a blue color. And so that's a synthesis chemical reaction that's occurring. And so, yeah, so the more protein you have, the more dye can react and then produce that blue color that you saw. That's why you have more blue in the presence of a greater amount of protein. So we've already answered these questions.
But why does dye cause a chemical reaction?
It's just the property of the dye. So the dye, this dye is reacts specifically. Oh wait, okay. I can't seem to find my controls. Sorry. Okay. So I don't know if did I share my screen?
No.
Oh, sorry. Let me just, okay.
There you go.
Okay. Sorry.
Bioluminescent proteins. Oh wait, no, never mind.
So yeah, so this is what's essentially happening is the dye specifically interacts with things only found on proteins. So there are particular amino acids that are only found, amino acid combinations that are only found on proteins that the dye will then bind to. And then when the dye binds to the particular amino acid regions that are found on proteins, it produces that blue color. And so it's just the particular properties of this dye. And this dye is called Kavasi blue. And they named it, it's called Kavasi blue because in the presence of protein, it goes blue. And so, yeah, so this is a protein dye. And so this is just one example of how chemical reactions, proteins are involved in chemical reactions. And so actually, I'll go back to the presentation. Our second example of a chemical reaction. We've done that. Oopsies. Okay. It's fluorescent proteins in different environments. And so I'm going to show you what happens when you put fluorescent proteins in different chemical environments. So what I'm going to show you now is what's going to happen to, where is my protein? Okay. Oh, so Adelis, how do I pin myself to the screen?
So if you go to the 3 dots, I can do it actually, and then spotlight for everyone. I just did it.
Okay. Okay, so I'm going to be working with this protein here. That's the green fluorescent protein. And so I'm just going to have a control tube of the green fluorescent protein that we can compare to. So this is, oh, let me see. It's really hard to focus the camera. Same protein, right?
Oh, two of the same protein.
Yeah. And so now I'm going to add this clear solution, which is an acid,
I was gonna.
to,
But don't acids dissolve protein? Wouldn't that like mess up the entire experiment or something?
well, let's see what happens.
All right, let's. Maybe my
So I've got my
correct.
acid.
Maybe it won't be correct. I'll never know until we figure out.
And then I've got my protein. And so let's start adding slowly to the side. Mix it in a bit.
Oh, I love this. I really do.
Still pretty much the same. Maybe a little bit of a color change, but let's see. Let's keep adding a little bit more acid.
To which one?
To our green fluorescent protein, this tube here. Okay. Just a little bit more acid. Okay, let's go here. Okay. So these are our two proteins. So this is the one that I'm adding acid to. Can anybody see anything?
The 2 looks a little bit brighter.
This one looks a little bit brighter. Let's add a little bit more acid and see what happens. Okay. Here we go. This could throw it over the edge. Okay, guys. Look what happened.
It's
happened to number 3?
It's clear.
It's just gone.
What happened to it?
It lost its ability to fluoresce in an acidic environment. So let's see if we can, do you think we can restore this back to this?
Well, if we took out acid, or if we added in more like water, I guess.
Well, water will just dilute it. How about we
add more protein?
Well, adding more protein might do something too, but
Chief.
How about we add something that is the opposite of an acid. Let's add a base. So this is my base. It's another clear solution. So this is just really concentrated base.
I don't know what it would do.
Okay, so let's just add a little bit of base and see what happens. And we have the fluorescence restored to the green fluorescent protein.
Oh, so the protein adapted really quickly.
Yeah, because it's a chemical reaction. And so
I thought it was just like, maybe I was wrong.
So green fluorescent protein, the way that it fluoresces is not due to a chemical reaction. But when you change its environment with chemicals, the chemicals then react with the protein to form their own chemical reactions, right? And so that's why when you add the acid, something happens. A chemical reaction takes place with the green fluorescent protein, which makes it stop fluorescing. And then when you add the base, this solution here, it changed the chemical environment that the green fluorescent protein was in, and then it restored back the fluorescence of the protein. So I'm just going to, so let's see what's going on there. So let's share my screen.
You're washing your
Okay. So what's happening? So when we added acid to the tube, we saw that as we added more acid, the protein stopped fluorescing. And then we added base to the tube, but we saw that the fluorescence was restored. And so what we're seeing here is an example of a chemical reaction called a replacement chemical reaction. And so what a replacement chemical reaction is you have A plus BC, for example, and then the C no longer is with the B, but joins together with the A and produces B. And so what happened is we have our green fluorescent protein. We added an acid, hydrochloric acid. And so what happened is the hydrochloric acid, the H part of the hydrochloric acid, attached itself to the GFP, which stopped the chromophore from performing its function of fluorescing, of emitting light. And so that's what we saw here. We saw a replacement reaction. And so then what happened with the base is the opposite. So the base essentially displaced the hydrochloric acid from the protein, restoring the ability of the chromophore to fluoresce. And so that's what's happening here. So what happens is in the presence of the acid, the H, the hydrogen from the hydrochloric acid, attaches to the nitrogen group of this aromatic ring here on the chromophore. And then when we added base, it neutralized it. So it removed the hydrogen from this nitrogen, returned the chromophore back to its neutral state, allowing the protein to fluoresce again. And so that is the end of chemical reactions today.